finish off making drinks for you, but I've heard coffee here is fantastic, so it'll be worth it for those of you having them. So first of all, just to say hello and kind of welcome to kind of all the new faces and kind of return faces here tonight. Just to tell you a little bit about Catholic Scientifique and how the format works. So we start off with a kind of 30-minute presentation from our two speakers here. We'll then have kind of a 10-15 minute break so you can grab another drink, things like that. And then we'll come back and we'll have a bit of debate and discussion that's going to be slightly different this evening because we've got a structured beginning to the debate, which I think some of you filling in a clipboard that's being passed around, I think we're going to look at kind of some of those issues to start off with. So yeah, a bit of different tonight. So to introduce our two speakers, so this evening we've got Dr. Catherine Mercer, who is a consultant in clinical genetics at University Hospital Southampton, and is also rare disease lead and education lead for the Wessex Genome Medicine Centre based in Southampton. Okay, and we also have Dr. Frank Rutland, who is the Senior Project Manager at the Wessex Academic Health Science Network, a company wholly owned by NHS England to drive the uptake of innovations into the NHS. He also has a PhD in genetics. So, without further ado, over to you guys. Okay, can you sit up there?
Well, thank you very much for that introduction. So I'm, as you said, Catherine Mercer, and I'm a clinical geneticist. So I specialise in seeing families with inherited diseases. And tonight I want to talk to you, as we can see, about the genomic revolution and something called the 100,000 Genomes project. And the discussion we're going to have will be around whether you would have your genome sequenced. And you've handed out the... Yeah, great. Can you hear me at this level? Is that loud enough? Yeah. Yes, okay. So, I think that we are in the middle of a revolution. And if you think about the Industrial Revolution, you may cast your mind back and think that that happened very quickly, whereas in fact it took about 100 years to happen. And I think the genomic revolution is similar to that. We're just at the beginning. So it was back in 1953 that Watson and Crick discovered the structure of the DNA double helix, albeit with the help of Rutland Franklin, who did the X-ray crystallography work. And I don't know where we'll be in 2050, which will be 100 years after the structure of DNA was discovered. So I don't know if we'll be living in these little futuristic towers. But I think that it's likely that life as a doctor, when you're looking after your patients, part of the information that you'll be accessing routinely will be not only somebody's blood pressure and their demographics and their age, but also down here will be something about their genome and the details of their DNA and how that relates to their health. That's what I predict we'll be doing. Now, just to take you back to what is a gene and what is a chromosome, all our cells, all our bodies are made of billions of cells, and inside every cell is a nucleus, and contained within that in humans are 23 pairs of chromosomes. So this is the nucleus with these sort of worm-like structures which are chromosomes. And if you take a single chromosome and unwind the DNA that it consists of, that stretches out to form this DNA double helix, and stretches of DNA are referred to as genes. And you've probably all heard of the term a gene. But just to ask, has anybody got an idea of how many genes there are in the human genome? Now, a genome is the word that we use for absolutely all of our DNA. Genes are the single smallest units of heredity that are recognised. So, has anybody got an idea how many genes we might have? Any offers?
100 million.
100 million, okay.
25,000.
That's a close guess. In fact, estimates vary, but it's about 20,000, which is quite humbling when you consider that's the same number of genes as a starfish has. And even worse, I was even more dismayed to discover that an onion has 4 times more genes than we do. tell us what they're all doing as plant geneticists. So, as I said, I'm a doctor and I look after people with inherited diseases. Now, when I first started, about 10 years ago, the best way for me to look at somebody's DNA in totality, so their entire genome, was to look down a microscope at their cells and look at all the chromosomes. And I could see if there was a whole extra chromosome present or a big chunk of chromosome missing. But if I wanted to know about one of those 20,000 genes, my job was to look at the patient and see the features that they had and think, well, which of our 20,000 genes has a mistake in it that's causing the problems that they have? And I'd ask for that one gene to be looked at, and it would take 3 months for the result to come back. And if I was lucky, it would be a yes, but otherwise it would be a no, and I'd try the next of the 20,000 doors, seeing if I could find out which single gene had a mistake in it that was causing the problems that my patient had. So that was quite a laborious process. But what's happened recently is that the technology has really changed and come on hugely. So where I used to ask for genes one by one to be sequenced, I can now ask for the whole human genome. So that's all 20,000 genes and the DNA in between them. I can ask for that to be sequenced overnight effectively. It takes about 48 hours to generate that data. And this is a picture of some modern day sequencers. And we've got there's a big factory essentially in Cambridge which houses tens of sequencers that are able to churn out this genomic data really, really quickly. And I don't know how many of you are familiar with the Human Genome Project. I suspect many of you have heard of it. So the Human Genome Project, when that took place, it took about a decade for the first person's entire genome to be sequenced. Now what that meant was we knew what order the 3 billion letters of genomic code were in. But it took about 10 years, it was an international collaboration, and it took billions of pounds. So that's sort of starting off here. Whereas now you can see the cost has come right down. And today we talk about the $1,000 genome, or pretty much the same as a £1,000 genome. So that's the cost in being able to sequence somebody's entire DNA. So I use this picture just to show how my job has changed really. So this was me 10 years ago going fishing with a single line and trying to pull that big fish out of the water and get the answer to the cause of the problems in the family that I was looking after. Whereas now, this is more like the answers that I get, which is wonderful having all this data, but the analysis of it is another thing altogether. You can imagine how you untangle which of the variants in the human genome are related to disease and which are just part of normal human variation. So the headache has changed now from getting the genes sequenced to interpreting the data. So a lot of the work that I do is on the neonatal unit. So I see lots of children, and I see the children particularly who are born perhaps with multiple congenital abnormalities that are unexplained, and I request investigations to try and find the underlying cause. And for some children, we have families and children in whom reaching a diagnosis has taken 10 years because they have conditions that are so rare that it takes a long, long time to reach the right investigation for them. Now, if we can use a genome to find that, we can sequence the whole genome to find out the cause, we can do that much more quickly, and we can prevent children from undergoing this long, drawn-out sort of diagnostic odyssey that can take years until the underlying diagnosis is reached. So, because of this change in the technology, and because of the appreciation that it's really valuable, in 2012, David Cameron announced the launch of the 100,000 Genomes project. Now this is a government-funded project, and it's under the umbrella of the NHS, although they set up a separate company called Genomics England to sort of be in charge of it. So the plan is to sequence 100,000 genomes. So it's not quite 100,000 patients, and that's because the project is split into two arms, and I'll explain in a minute why it's not quite 100,000. But it'll be 70,000 patients will be involved across England, and we've got 13 sort of main hub hospitals. So I'm based in Southampton, where our Genomic Medicine Centre is, but I recently did a clinic in Bournemouth as well, and we're recruiting patients locally as well. The other important thing about the project is that, as well as sequencing the DNA to try and find a diagnosis, there are also links with industry partners, and it's important for any patients who are enrolled to know that. And the reason that that's being done is that we're hoping that with this genomic information, we can begin to develop new drugs, for example, and we can begin to find out how somebody's genomic architecture influences which drugs work for them and which ones don't. And the NHS isn't in the process or doesn't develop new drugs. This is largely done by pharmaceutical companies, which is why the data will be shared, but it will be shared anonymously, but companies will have access to be able to use that information. So I'd be delighted if I heard that any of you had seen this poster, but in Bournemouth we haven't rolled them out just yet. This is the poster that heralds the news that in Southampton, Basingstoke, Portsmouth, Winchester, we're part of the 100,000 Genomes project and we're recruiting patients. So what's this project all about? Well, the framework upon which this is based is, first of all, one of the most important premises is that the programme's based on ethically transparent consent that's given by patients before they're enrolled. And patients, once they consent, they can withdraw that consent at any time. And we go through in quite a lot of detail. It takes about 40 minutes to be entered into the project to give the full details of what that involves. We're hoping that benefits will be brought to patients across the NHS, and we're hoping to be one of the first healthcare systems in the world that really embeds genomic technology into mainstream healthcare. We're hoping to discover some new genes along the way. So, although the Human Genome Project told us where the genes sat, it didn't tell us what the individual genes do. So we're gradually learning one by one what each of these 20,000 genes do, and every week there's a new gene published that we learn is associated with a particular disorder. So hopefully with the gathering of so much data, we'll be able to discover new genes. And this last picture refers to the stimulation of UK industry. Now, genomics, I think, is an ideal industry for a small place. This is information rich, and you don't need much space to do the wet work, the actual sequencing of the genomes, but you need a lot of brains in bioinformatics and interpretation in order to really get the true benefit of this data. So having this upfront investment now in the 100,000 Genomes project, I think will be a kickstart to our genomics industry. So this is a picture of England, and in terms of where we are recruiting patients. So patients, as I said, are being drawn from all over England to enter this project. And this just refers to the London centres. There's 3 centres in London. There's Southampton. The nearest big centres are Exeter, Oxford, and now Bristol, actually. But essentially, the idea was to offer equal patient access from across the country so that all of those eligible can be enrolled into the project. So I mentioned that the project is split into 2 halves. So there's a rare disease arm to the project and a cancer arm to the project. Now, I'm primarily involved in rare diseases, but the interesting thing about a rare disease, so rare diseases are classified as diseases that affect 1 in 2,000 or fewer people in the population. And 80% of these rare diseases are genetic. A large proportion affect children. So although each individual disease is rare, there are lots and lots of them, so that about 1 in 17 of the population have a rare disease. So in this room, maybe 3 of us would have a diagnosis of a rare disease. So individually, they're not common, but if you look at all of the rare diseases together, they affect a large proportion of the population. So the other side of the project is looking at patients with newly diagnosed cancers. Now, in the rare disease arm of the project, we'll be looking at the DNA from the person who's affected with whatever condition it is that we're looking at. And I can talk you through what types of diseases we're recruiting. Now, with the cancer side of the project, patients are being recruited who have a new cancer diagnosis. And the relevance of the genome to cancer, well, we know that all cancers are actually, they're a disease of DNA in a way, in that all a tumour is unregulated cell growth. Now, cell division is under the control of our DNA, but when you have faults in your DNA that you accumulate through ageing, say, which is why cancer is typically, or more typically, a disease of older age, when you get mistakes in your DNA that are accumulated through ageing or excess sunlight or cigarette smoke or poor diet, for example, the DNA is damaged, and then the cells that should reproduce from that genetic code don't have the right recipe, if you like, to work from, and so the cells reproduce in an unregulated way and form a cancerous tumour. So for the cancer side of the project, we'll be looking at the DNA that a person's born with, which is called their germline DNA, and we'll be comparing that DNA to the genome or the DNA in their tumour, and seeing what it is about the DNA changes that have resulted in that cancer. And again, we're hoping to be able to stratify treatment based on what we find. And in fact, we're already doing this to an extent. So there are some lung cancers, for example, where the tumour is routinely tested for a particular genetic mutation, and if positive, patients have one particular treatment, and if negative, they don't have that treatment, which we know will never work, and we spare them the side effects associated with that, the unrealistic expectation, and the time wasted having a treatment that was never going to be suitable. So, as I say, the rare disease and the cancer arm of the project. So this is a slide. It does duplicate a little bit of what I was saying about the scale of rare diseases. So I said that rare diseases affect about, 7% or 1 in 17 of us, and there are perhaps up to 8,000 different rare diseases. So as a geneticist, I know about some of them, but I confess I don't know about all 8,000 rare diseases. And this little picture down here with the text tells us that rare diseases are responsible for 35% of deaths in the first year of life. So really important for the newborn population. So for patients who are going to take part in the project, and this picture's split up into rare disease and cancer. So for rare disease patients, and the type of problems we're looking at. So I'm a geneticist who also specialises in cardiac genetics, and within the cardiac field, we're looking at things like familial hypertrophic cardiomyopathy, where the heart's abnormally thickened, or there's a condition called long QT syndrome, which runs in families and predisposes to unusual rhythms of the heart. We're looking at many childhood conditions, so multiple congenital abnormalities. We have a lot of patients enrolled with intellectual disability, trying to find out the cause for that. So the rare disease patients, we take DNA from the patient, and then ideally we take it from both parents, particularly if we think it's a recessively inherited condition. And essentially what's done is you compare the DNA of the affected patient with that of their unaffected parents, and you effectively, it's a complex spot the difference to see which changes are in the child. We also look at them in terms of their bioinformatic properties, if they've been reported before in other individuals with a similar condition. And then for the cancer patients, as I said, we take DNA from the germline, so from the blood, and then from tumour samples as well. Now, we all have mistakes in our DNA, or little alterations. They're part of what makes us human, and we have to try and tease out which are the ones that are important in disease and which are just part of normal human variation. And we can only do that if we're able to couple the sequenced genome with the health records of a person. So as well as people being enrolled into the project giving a blood sample, we also need to have consent for them that we can also look at their medical records and see, get the details of the disease that they have and perhaps the pattern of that disease in their family. So we look at the medical information, the genome, and then we try and match the two. So have mistakes in this particular gene been associated with these particular features before? So the sequencing, this slide just says the information is analysed, and this is sequencing data. And the sequencing is being done in effectively a sequencing factory outside Cambridge. I don't know if you've been there, but it's at the Sanger Centre, and there are rooms full of DNA sequences. So the sequencing is being done there, and then they'll send back a short list of genetic or genomic variants to us in our local centres for us to interpret more fully. So in terms of what we're telling participants about the results that they'll get, well, as a clinician, the most important thing is that the question that the family are asking me, so why does my child have this disability? Why do people in my family, why do lots of people have unusual aneurysms of the heart, for example? Why is there deafness affecting lots of people, or cataracts? The most important thing we'll be telling them is the answer to that initial question. So what's the cause for this presenting problem? But we're also offering patients the opportunity to learn about some what's called additional findings. So patients who are enrolled into the project have a chance to have additional variants looked at for genes that cause conditions for which medical management is available. So most of these are cancer predisposition genes. So breast cancer, for example, you may have heard of the BRCA genes that predispose to breast and ovarian cancer. There are also bowel cancer predisposition genes. So individuals can opt in and say, yes, I don't only want to know the answer to the primary question, I also want these additional findings fed back. That's optional. And lastly, if people taking part are thinking of extending their family, they can also be told if they're carriers for some common conditions. So, for example, cystic fibrosis. About 1 in 20 to 25 of us are carriers for cystic fibrosis. And of course, if we choose to have a family with someone else who's a carrier, we run the risk of having an affected child. So people can find out if they're carriers for common conditions. So we thought it would be helpful to, as well as me telling you the sort of framework of the project, to learn a little bit about what the patients who are involved in the project, what the participants have been telling us. And Frank's going to talk us through a couple of, or guide us through a couple of patient stories.
Thank you, Catherine. So I'm Frank Ratcliffe. I'm a project manager from Wessex Academic Health Science Network. That's a small piece of the NHS that exists to help drive innovation. I've actually got 3 patient stories to go through. 2 of them are patients presenting themselves, and then the third one is a little bit more detailed, and I've got a couple of slides on that. Can I check it at the back? If you can't hear me, wave, and I'm about to play some video. If you can't hear that, wave as well. Thank you.
Know anything about Musa and some people.
Technical support's brilliant.
Absolutely. They said you could go home now, no problem with Musa. Just we need to do like tick boxes, check his health in general, and they did check his oxygen level in his blood, and they were about 88%. And they say, oh, there's something wrong here. His general health looks okay, but then after they did an echo test in the intensive care, they found loads of problems. The consultant sat down and tried to explain what's happening to Musa. And he said, Musa's heart has got a big hole. I think it was about 22 mil out of 30 mil of his whole heart. And then I said, what else? Is that all? He said, no, there's another problem. It's called transposition great artery, which means all the bloods, they go to the wrong place. The oxygen blood and non-oxygen blood just goes to the wrong place. I said, is there any more problems? He said, yes. Something called double outlet right ventricles. Means again that all the tubes goes to the wrong place. And last thing he said is pulmonary stenosis. The tube comes 13 mil, goes to 3 mils. So it gives a lot of flood to the lung with the blood. So he can't stand it. It was shock for us. And I said, is this problem, is it repairable? Is it kind of curable? He said, I'm afraid it's not. Our Prophet, peace upon him, Muhammad, peace upon him, says, if you have one day in your life and you have a plant, you should seed the plant, or you should plant it. Maybe you don't make benefit from it, you get to die, but somebody else will make benefit from it. On that basis, I think if there's anything I could do to help my child, others might be beneficial to my child from this research, might not be beneficial, but I'm sure it will be beneficial for people, like maybe years to come. Some people are worried about that data will be exposed to, and will be, worried it might be, exposed to public, or it might be used by some commercial companies. As far as I'm concerned, commercial companies doesn't bother me. They make benefit fine with me, as long as they make benefit to humanity, to people like Musa, my child, then I'm okay with it. Personally, I don't like my data to be exposed to be public domain. But if they've been contained within research team and other researchers, I'm a researcher myself, so I could understand that. I think it's okay, no worries about that.
I've obviously seen that video a few times, and it still touches me, because that's a family who...
My name is John Marchmont. I had an aortic.
Thank you. So that's a family who already know that there's no help for their child. And what the guy said, I don't know how audible it was at the back, was if you have one day, then use it to plant a seed. Where's the next?
We go back to presentation.
Dissection some 4 years ago. An aortic dissection is a split in the aorta. I discovered that I had one when I found that I was suffering from a
So this video.
Yep.
Okay. Can we go?
Full screen.
My name is John Marchmont. I had an aortic dissection some 4 years ago. An aortic dissection is a split in the aorta. I discovered that I had one when I found that I was suffering from a severe pain in my chest, and eventually made my way to Hope Hospital, where after a couple of questions from the doctors, they established that it was an aortic dissection I was suffering from. My father died some 14 years ago following an aortic dissection, and then obviously I had one 4 years ago. Having been contacted by St Mary's with regard to the Genome project, we did some research into how my grandfather had died. We always believed that he died as a result of a heart attack. But when we actually uncovered his death certificate, we found that he too had died as a result of an aortic dissection, meaning 2 out of 3 in the family had died as a result of it. I have 2 sons, one aged 30, one aged 27, and obviously I'm concerned that with the family history that they may also carry the faulty gene. I'm hoping that as a result of the test that they've carried out on myself, that this faulty gene certainly in my family will be identified, and that it will benefit other families who may be suffering from the same problem. If diagnosis is possible, then it would obviously certainly put my wife and my mind at rest, in that something could possibly be done to prevent the same thing happening to my two sons in the future. I'm more than happy and more than willing to help, and that's how I personally see it, as helping others as well as my own family. I've certainly got nothing to hide. I've got nothing to be ashamed of, that I've had an aortic dissection, and hopefully if my experience can help other people come forward and join the project, then that would be great.
How do I stop it? Ah,
So
there we go.
click back onto it. So if we get out of that one, and then
Fantastic. Thank you. So John obviously had a narrow escape, and I think he's got clear concerns that the same gene could affect his immediate family, and he's got no problem sharing his data. I can't imagine it would make a difference to any preventative treatment for his sons, but he's just imagining future generations down the line and the help that he could give in preventing that being spread. The last patient story is Jessica. So I've actually seen Jessica's mum present at a national meeting. Jessica was suffering from symptoms of severe epilepsy, so seizures and some developmental difficulties. She'd had multiple investigations. She'd been around multiple consultants, multiple hospitals. Some of her investigations are probably slightly intimidating. If any of you have been inside an MRI scanner, I wouldn't want my small children to be in there. Lumbar puncture is more than intimidating, it's uncomfortable. And there was still no clear diagnosis. The treatment she was on was increasing doses of anti-epileptic medication. So these are difficult medications to take with significant side effects. It was not resolving her seizures, and the only suggestion to go forward was to increase the dose. And at that point, Jessica was offered the opportunity to join the 100,000 Genomes project. So obviously the questions at hand are, can you get a diagnosis, can you get a treatment? And at that point, Jessica was an only child, so there's a significant question for her parents. Do you risk having another child? If you have another child, is that a risk that you'll be in the same position? As Catherine said, what's involved is a blood sample from Jessica and from her parents, and then essentially you're playing spot the difference because the parents don't have this condition. Three billion base pairs, 3 billion letters, 3 billion ATCGs. When we stop for drinks break, we'll pass round what DNA looks like. The other book, I'm afraid, is propping up the projector. So the two books together are the smallest of our chromosomes. It's chromosome 21. It's printed, double-sided, narrow margin, in 4-point font, and it takes 2 books. To print the whole genome the same way would take 130 books, but my knapsack isn't big enough. And it's just interesting to see because there's no punctuation, there's no start-stop, there's no you are here. So to find a difference is quite a challenge. To find a significant difference and understand which single letter change is clinically relevant comes back to the point that Catherine made earlier about the volume of data. if you get here, and if you did go to Specsavers, you might find you can see in here the genes are in capitals and the bits between the genes are in lowercase. But obviously if you just get raw sequence data, even that isn't there. Okay? So have a look at this, don't nick the one under the projector. So 3 billion base pairs. Compared to her parents, there were 6,400,000 differences. Okay, so that's probably about normal. Between normal, people of the same ethnic group, I think there's normally about 3 million differences. So that's between her 2 parents. In this needle in a haystack, there were 700,000 that were classified as rare. She obviously had a rare disease. There's 3,000 changes that would affect a protein. So genes make protein, and fundamentally we are all protein, because if we're not actually protein, we're things that are made by enzymes which are protein. So it's all about protein changes. Of the 3,000 that would affect a protein, 67 were not shared with her parents, and 1 had been previously linked to symptoms like hers. It's a deletion in a gene that makes a protein that transports glucose across the blood-brain membrane. So without that transporter, you don't get glucose into the brain, and so effectively her seizures were episodes of hypoglycemia, as you would get if you were type 1 diabetic with too much insulin. Only 500 cases known globally, so extremely rare disease. And I think if you think from a clinician's point of view, you're probably never going to see one of these in your career. If you do, you'll probably only see 1. So without some kind of database and scalability, how do you find a useful treatment? Well, the brain can make its own glucose. All our bodies can make their own glucose out of fats. So the suggestion, and there's some evidence behind this, was a low carbohydrate, high fat, ketogenic diet as an alternative energy source to carbohydrate. And the small print here says, do not try this at home. And there's a very positive outcome. So, firstly, there's no genetic change which is shared with her parents. So for her parents, having another child wouldn't have an increased risk of that child having the same condition, which is obviously a huge piece of news for that family. Jessica's symptoms are much better controlled on a ketogenic diet. So she's not symptom-free, it's not a complete transformative magic wand cure, but perhaps if she'd had that diet from birth, if this had been picked up 7 years ago or 4 years ago when she was first diagnosed with a problem, then some of the developmental issues might have been prevented. We don't know because we can't wind back the clock. And obviously, as we saw from Moose's story and from John, not every individual is going to have this miraculous outcome, but it does show what can be done. And it shows what can be done. This was the Industrial Revolution, moving to high-scale weaving, away from little cottage industries with looms. And these are the mass sequencers which transform the speed at which this data can be produced and then analysed. And so that really brings us to the end of the first half and opens the question, would you have your genome sequenced? Would you see it as an opportunity to take control? Or would you rather bury your head in the sand? It's a very difficult question. We passed around a little clipboard earlier as we were accumulating. Hopefully that's done the rounds. And I am going to confess that we don't have a free genome to sequence. Sorry. But what we are going to do is talk through, hopefully, some of the people who said yes, they'd like to have their genome sequenced, why that is, and for those who said no, what their concerns would be. So we're going to have that as a discussion, and after that, we'll collectively take any questions. Okay? Fantastic.